Welcome to the deep dive. If you've been following the incredible promise of personalized medicine, especially when we talk about designing vaccines for things like cancer, we've kind of hit a wall.
A huge wall. It's the critical bottleneck, really. We have this, information overload, but a severe knowledge deficit.
And today we are diving into sources that detail ImmunoStruct, which is a brand new deep learning model that was, developed specifically to solve this exact problem.
That's right. The whole mission is to find epitopes.
Right. And for anyone who needs a quick refresher, epitopes are just these tiny short fragments of proteins. They're what we use in these advanced vaccines to basically train your immune system's killer T cells.
Your CD8 plus cells.
Exactly. To train them to recognize a threat, whether that's a tumor or a virus.
But here's the massive challenge. Finding an epitope that's actually immunogenic, one that really triggers a strong T cell response, is notoriously difficult and, expensive.
And the old computational methods, they were relying on basically one thing, right? Just the amino acid sequence.
Almost entirely.
And when you only use the sequence, you're pretty much just guessing at the outcome. The scale of that failure is, it's staggering.
It really is. Our sources confirm that historically, only 2 to 6% of peptides that were predicted by computers
Only 2 to 6%?
actually resulted in a genuine, measurable immune response in patients.
Wow. When you're talking about clinical trials, that kind of success rate is it's a monumental waste of time and money.
And for the patient, it's a waste of critical time. So we desperately need better tools that can look beyond that simple blueprint and, understand the actual 3D architecture of the problem.
Okay, so let's unpack this. ImmunoStruct is the answer here. It promises better accuracy, and crucially, it's interpretable. It does this by using what's called multimodal data sequence, structure, and biochemistry.
But before we get into the AI itself, we should probably lay out the foundational biology that makes this whole thing so tricky.
Good idea.
It all comes down to the major histocompatibility complex, the MHC system.
Think of the MHC class I system, which is on pretty much every cell in your body, as a kind of display stand.
A standardized display stand, exactly. It holds up these little peptides, these epitopes, on the cell surface for inspection.
And the inspectors are the cytotoxic CD8 plus T cells. They're the immune system's executioners, really.
Right. So a T cell is patrolling. It comes past a cell, and it uses its T cell receptor, or TCR, to sort of shake hands with that MHC peptide complex.
Handshake. I like that.
And if that handshake feels wrong because the peptide signals cancer or virus, the T cell pulls the trigger.
So the problem with the old sequence-only models was that they were only predicting one tiny part of that process.
Precisely. The whole process is incredibly complex. You have antigen processing, then the peptide has to bind into the MHC groove, then it has to be presented on the surface, and only then do you get that final critical TCR recognition.
So a sequence-only model might say, yep, peptide A should bind really well, but that's just step 2. It completely misses the whole third act.
It misses how the peptide sits in that cradle, what its final shape looks like, and whether that shape is even recognizable to the T cell.
So that's the core flaw. The physical three-dimensional structure of that whole complex is what dictates the outcome.
Yes. You could have 2 peptides with similar sequences, maybe even similar binding scores, but one might bulge out perfectly for that TCR to grip it, while the other just lies flat, totally invisible.
And that distinction, the visible bulge versus the invisible flat one, that's why we have those dismal 2 to 6% success rates.
It's a geometry problem.
Yeah.
Not just a sequence problem.
Okay, so this is where ImmunoStruct gets really interesting. Its architecture uses 3 separate specialized neural networks. Let's start with the first one, modality 1, the blueprint, the sequence.
So for the sequence, ImmunoStruct uses something called a variational autoencoder, or a VAE.
Okay, why complicate things with a VAE? Isn't a sequence just a string of letters?
It is, but simple encoding is too rigid, too literal. A VAE performs a really sophisticated kind of compression. It takes the whole sequence and maps it to a compact 32-dimensional, what they call a latent space.
A latent space, so like a map.
Exactly. Think of it like a complex neighborhood map. Peptides that are functionally similar, maybe they're just 1 or 2 amino acids different, they get placed right next to each other on the map.
So it's creating a kind of semantic continuity. The AI learns that sequences that act alike belong together, even if they aren't spelled exactly the same.
You got it. And that makes the model really robust, and it lets it learn these essential sequence motifs much more effectively.
Okay, but since we know sequence alone failed, that brings us to the really critical part, right? Modality 2, the shape.
The structure, yeah.
How do they even generate the 3D data for this? I mean, these structures are notoriously hard to get.
This was a huge undertaking. They used the high throughput power of AlphaFold 2, the big protein structure prediction tool, to actually fold the peptide right into the MHC protein.
Wait, so they didn't just use existing libraries. They generated a whole new data set of predicted 3D structures just for this.
A massive inconsistent one, yes, which is key for training.
So how do you feed a 3D shape into a neural network?
You turn it into a graph. Every amino acid becomes a node, and that node knows its identity and its 3D coordinates. And the chemical bonds between them, peptide bonds, hydrogen bonds, all of it, those become the edges connecting the nodes.
And that graph gets processed by this EN, equivariant graph neural network, the EGNN.
The EGNN.
What does equivariant mean here, and why is that so important?
Equivariance is basically where they build the laws of physics right into the AI. Imagine you're looking at one of these 3D complexes on your screen. If you rotate it at 90 degrees,
right,
the underlying physics, the binding energy, the stability, none of that has changed.
Of course. It's the same object, just a different viewing angle.
Exactly. And the EGNN layers guarantee that the model's prediction stays consistent or equivariant, no matter how you rotate, translate, or flip that structure in 3D space. It learns the inherent physics of the shape, not just an arbitrary snapshot.
Okay, that's brilliant. So we have the sequence compressed by the VAE and the 3D shape understood by the EGNN. What's the final piece? Modality 3, the physics.
That's the straightforward biochemical properties, things like molecular weight, charge. But the most important one they included here is a structural feature called the solvent accessible surface area.
SASA.
SASA, right. That's basically a measure of how much of the peptide is exposed to the outside world.
How much is available for that T-cell receptor handshake?
So you need structure for stability, but you need surface area for recognition.
Precisely. So you take these three embeddings, the 32D sequence, the 64D structure, and the 8D biochemical vector, and you combine them.
And it's not just a simple mix. This combined 104-dimensional vector goes into a multimodal, multi-head attention mechanism. So what's this engine room doing? This is where the synergy really happens. The attention mechanism dynamically assigns weights. For any given peptide, the model basically asks itself, okay, for this one, should I pay more attention to the structural clues or the sequence clues?
So it can learn to focus on what matters most for each specific case.
Yes. And they boosted this whole process with a smart training strategy called transfer learning.
They didn't start from scratch.
No, they leveraged basic immunology. They first pre-trained the model on predicting foreignness, just how different a peptide is from known human proteins. This taps into the whole concept of self-tolerance.
So the model gets a basic intuition for self versus non-self before it even starts learning the much harder task of immunogenicity.
Exactly.
Okay, so let's pause for a second here. We've established the foundation. ImmunoStruct is this unified multimodal framework. You have the VAE for sequence, the EGNN for physics-aware 3D analysis, and SASA for T-cell accessibility.
The key takeaway is that shift in focus.
So a question for you listening. If the traditional methods only ever used sequence, what core immunological principle, that three-dimensional handshake between the MHC peptide and the T-cell receptor, did they inevitably miss?
They missed the part that actually dictates T-cell recognition. Sequence can get you a strong grip that's binding affinity, but it's the structure that gets you the perfect grip and the perfect angle for the T-cell to land and activate. It's the difference between a peptide just sticking to the cell and a peptide waving a giant flag that says, attack me.
Let's pivot to the highest stakes application, cancer. Because with cancer neoepitopes, you're often talking about a difference of just a single amino acid from a normal self-protein.
And the immune system is, by default, tolerant to that normal self-protein.
So how on earth do you teach a model to spot that one tiny change that's enough to suddenly break tolerance?
This required a really clever cancer-specific innovation. They call it the cancer wild-type contrastive loss. It basically bakes the idea of immune tolerance right into the AI's math.
Okay, how does that work in that latent space, that map we were talking about?
So think of that latent space like a big gallery wall, and every peptide is a picture. Now, if a mutant peptide is non-immunogenic, it means the immune system tolerated it. It's functionally the same as the self-protein.
Right.
So the loss function tells the model, pull those two pictures closer, put the non-immunogenic mutant right next to its normal wild-type version on the wall.
It's forcing the AI to understand the concept of tolerance.
Yes. But if the mutant is immunogenic, it succeeded in breaking tolerance. The loss function then says, push them apart, Shove that immunogenic mutants picture as far away from the wild type as you can.
That's a brilliant way to do it. It forces the model to learn the specific, subtle differences that are required to break self-tolerance.
And the results were state-of-the-art. On the big infectious disease dataset, IEDB, ImmunoStruct, got an AOROC of 0.882, which is great. But the really critical number was the AUPRC, which was 0.696.
Okay, let's talk stats for a second. Why is AUPRC so much more important than UROC in this case?
Because predicting immunogenicity is a wildly imbalanced problem. I mean, 90-something% of all possible peptides are duds. They do nothing.
So a model could just guess non-immunogenic every time and have super high accuracy, but it would be completely useless.
Exactly. AUPRC, on the other hand, harshly punishes false positives. And in vaccine design, a false positive means you're wasting millions of dollars and months of time testing a dud.
And the old models, they were, what, in the 0.2 to 0.4 range?
Often, yes. So going to nearly 0.7 is a huge leap in clinical utility. You're finding much better signal in the noise.
But the real payoff of using structure, I think, is the interpretability. The model can actually tell us why it made a prediction.
This is the power of the attention visualization module. They could literally see where the AI's eyes were focusing. And on the peptide itself, the results were incredible.
Where did it look?
The attention was really enriched at the TCR-facing positions, the residues that physically stick out, available for the T-cell to touch. And, crucially, the model learned to ignore the anchor positions that just hold the peptide down deep inside the MHC groove.
So the AI figured out on its own that it shouldn't just look for what makes the peptide stick, but what makes it visible. It learned the geometry of a T-cell handshake.
It confirmed known biology completely from the data.
Okay, let's get to the KRAS case study. This is an oncogene that drives so many common cancers. They tested 2 mutants, G12V and G12D.
And ImmunoStruct predicted that G12V was significantly more immunogenic than G12D. And this aligns perfectly with experimental data from actual clinical vaccine trials.
But here's the part that is just mind-blowing. When they looked at why the model made that choice, where did the attention fall? Was it on that single mutation site at position 12?
Surprisingly, no. The big difference in the model's attention was concentrated way down at the C-terminal end of the peptide.
Wait, hold on. You're saying the single amino acid change at one end of the peptide caused a ripple effect, a structural change far away from the mutation itself.
That's exactly what the data suggests. A complex, distal conformational shift.
So the thing we thought was the red flag, the mutation itself, wasn't the actual flag. The mutation created a subtle bulge somewhere else entirely, and that is what the T-cell actually recognized.
A subtle structural determinant that was completely invisible to every sequence-only method ever created. It proves, without a doubt, that this is a 3D problem.
So if a single mutation in a cancer gene can cause this distant structural shift that changes the entire immune response, how does that fundamentally change how we need to screen for cancer therapies?
It means we can't rely on proximity anymore. We have to integrate predicted structure into every single step. We need tools that can map a genetic change to its 3D functional consequence. Otherwise, we're flying blind.
And this wasn't just in silico. The model stood up to real-world validation.
It did. On SARS-CoV-2 epitopes, it showed 79% accuracy against lab-based T-cell assays. But the really compelling part was the clinical validation with cancer patients.
Tell us about the survival prediction. This is where the rubber meets the road.
They took a group of 70 cancer patients on immunotherapy and used ImmunoStruct to predict their total load of effective epitopes.
And the result?
The model score was a very strong predictor of both overall survival and progression-free survival.
With a hazard ratio of 0.54.
Which is massive. It means patients in the high immunogenicity group were roughly half as likely to experience progression or death.
Wait, so this AI structure model is a better predictor of survival than just counting the total number of mutations with something like tumor mutational burden or TMB?
That's what the data showed. It's a huge shift. It suggests that the quality of the mutations is far more important than the sheer quantity. And for the first time, we have a tool that can accurately measure that quality.
That is quite a journey. From a biological bottleneck to an AI solution that can actually help guide clinical decisions. So to summarize, ImmunoStructs prove that combining sequence, structure, and biochemistry unlocks this critical predictive power.
Of course, it's important to remember the limitations. The training data is still messy, from different lab protocols. And crucially, it couldn't incorporate T-cell receptor information yet. The human TCR repertoire is just astronomically diverse.
Those are big hurdles for sure, but it doesn't diminish the achievement. This deep dive really shows how AI is moving from just prediction to real functional insight. It's bridging computer science and immunology in a way that directly impacts vaccine design today.
It gives us a whole new lens for how the immune system operates at a structural level.
It really does. So here's a final thought for you to chew on. If AI can reveal that a tiny conformational bulge miles away from the original mutation is what dictates whether a T cell kills a cancer cell, what other subtle structural determinants in biology are we completely blind to right now? Things that could totally revolutionize how we discover all kinds of new therapies. We might be standing at the dawn of structure-first medicine.